Well, welcome everyone. I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I would also like to pay my respects to elders past, present and emerging. Well, good afternoon and welcome to the June FightMND Scientific Seminar. My name is Sarah Bennett and I'm the Research Coordinator at FightMND. We hold these seminars monthly on the fourth Tuesday of the month, and in May we heard from Professor Julie Atkin from Macquarie University. Julie spoke to us about a new protein linked to MND that may be developed as a non-invasive biomarker of disease. If you missed out on this presentation, it is now available on our website, so you can catch up at a convenient time. So following the talk today, we are leaving plenty of time for discussion and questions. So if you would like to ask anything, please do use your reaction button to raise your hand, and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, you can also type your question into the chat where our team will be monitoring and read out questions on your behalf. Please note that the chat function is only enabled if you are using the app-based version of Teams and does not work in the web-based version. Please ensure you introduce yourself and where you're from before asking your question. So now to our presentation for today. Larry Steinman is a professor of neurology and neurological sciences at Stanford University in sunny California, USA, where he has been a member of the faculty for 44 years. He came to Stanford in 1973 after graduating from Harvard Medical School and has since established himself as a leader in the field of neuroimmunology. For his role in discovering that alpha-4 integrin is key to T and B cell entry into the brain in neuroinflammation, Professor Steinman was elected to the National Academy of Sciences and to the National Academy of Medicine. Professor Steinman has also founded and served on the board of several pharmaceutical companies and currently serves as chairman of the board of Pasithea Therapeutics. He joins us today to tell us about a neuroinflammatory therapeutic candidate for MND that he is developing. A very warm welcome from Australia, Larry, and thank you so much for joining us today.
Thank you very much, Sarah. And first of all, I would really like to thank the Fight Motor Neuron Disease Philanthropy for all the hard work you're doing to help us in research get some progress in the field and eventually wipe this disease out. It's an aspiration that I'm sure we all share, but without funding, it's going to be hard. And I really appreciate the grant that we received, and I'm going to share the data with you. So a little bit about my background. I'm a neurologist, and for most of my career, I've been focused on another disease that has the word sclerosis in it, and that's multiple sclerosis. And the title of this talk is back to an old playbook. So along the way, in the 44 years I've been at Stanford, we were very interested in how inflammatory cells get into the brain in multiple sclerosis. And it turned out from a series of simple experiments that one of the integrins, alpha-4 integrin, was quite important. And that led to the first monoclonal antibody therapy developed for MS. It was a therapy that had a liability. If your immune system cannot get into the brain, inflammation won't occur, but the viruses can take over. And initially, that therapy was plagued by the first group of patients getting progressive multifocal leukoencephalopathy. But a risk mitigation test was done, and now we don't see that with the anti-alpha-4 because we screen patients who are at risk. So what I thought I would do as my research progressed, ALS has always been one of the most disturbing of all the neurologic diseases that I see when I put on my white coat and go to the clinic. So I was very interested in looking at integrin biology in this disease. Let's go to the next slide, please. A couple of disclosures. Along the way, the discoveries made at Stanford were licensed to Pasithea Therapeutics, a company I co-founded. And so far we have 2 patents on the alpha-5 technology, one issued in Japan and one in the European Union. Next slide. So again, the playbook goes back way before I was looking at lymphocyte homing, and I was at an art exhibit at New York University where they had drawings from Ramon Cajal, who discovered that neurons were the key cells in the nervous system, and his students discovered glia. But he was a wonderful artist and photographer, and he showed pictures that he saw in his microscope that he sketched of lymphocytes going through endothelium. So about 20 years after we discovered the alpha-4 story, somebody asked me to write a review, and the artist drew a picture that's shown on the right. But had I known about Cajal's picture, I just would have had his picture as the banner piece. And I wanted to show the next slide. Let's go to the next one, Sarah. That there's a cliche that's very true, that we stand on the shoulders of giants. So over on the right are 3 giants of the field of integrin biology. They won the Lasker Prize last year, and they might get another award one of these days. But the picture on the left is very interesting because these are people who are more like you and me, ordinary people. They happen to be street entertainers, but the giant Ramon Cajal took the picture because he was very interested not only in his sketching, but in photography. So that I would call standing on the shoulders of gifted street entertainers taken by a giant in science. Now I'll turn to some science. Next slide, please. So this is the group that did all the hard work. Bahara Ajami was a postdoc and then a fellow in my lab for about 9 years. And I'll talk about her work that led to the identification of alpha-5 integrin on macrophages and microglia. And her first postdoc was Aude Chiot, who came from Paris, where she had already done work on macrophages in ALS. In the middle pictures are Dennis Dixon and Shanu Romer. They're at the Mayo Clinic in Jacksonville, and they looked at 132 autopsies. There's a lot of material at the Mayo Clinic because it's one of the leading referral centers in the United States. And they really showed something quite dramatic about the spatial zonation of alpha-5 integrin in the spinal cord and brain of individuals with ALS. And then on the right are 2 gifted scientists who worked on this at Pasithea, Mike Levitin and Lisa Reiner. Next slide, please. So Bahara Ajami's paper identifying alpha-5 integrin on macrophages and microglia was published in 2018, so already 6 years ago, where she used a technique developed at Stanford by Gary Nolan called CyTOF. It's a combination of mass cytometry and flow cytometry, where heavy metals isotopes are used to discriminate different antibody markers. So she did an immense job looking at experimental models of classic neuroinflammation and classic neurodegeneration. I say classic because the quintessential model of neuroinflammation is called the experimental autoimmune encephalomyelitis model. And that was the model we used initially to show the importance of alpha-4. And then she studied two other models, the Huntington's disease model and the R62 with an expanded trinucleotide repeat, and the SOD G93A mouse. So the whole idea there was I wanted to know if there was a boundary between what we call neuroinflammation and neurodegeneration. And the more I look into this situation, I don't see a very clear boundary, but we've learned a lot. So anyway, she used mass cytometry, and she showed that CD49E, that would be alpha 5 integrin, was increasing in the spinal cords of the SOD mouse as the disease progressed. Let's go to the next slide. So I'll show you work that alpha 5 beta 1 is overexpressed in both human and mouse ALS. And it's a well-characterized target. And actually, in cancer studies, an antibody to alpha 5 beta 1 was taken into the clinic, and over 100 patients were treated with a very good safety profile. It didn't work so well to reduce the spread of the cancer. And the drug that was taken most deeply into the clinic was called volociximab. And it, as I said, went to phase 2, and there was good safety signals. Now, there's 3 FDA-approved monoclonal antibodies. One is the one I talked about, natalizumab Tysabri, alpha 4 beta 1 is what it targets. And Tim Springer showed that alpha 4 beta 7 was critical for homing of T cells and B cells to the gut in inflammatory bowel disease. And ReaPro was an FAB targeting a different integrin for stent stenosis. So integrins are druggable targets, and that's one of the attractions of studying this in a disease like ALS. I'll show you in coming slides that the alpha 5 beta 1 is on macrophages that are coming in from the periphery and also directly attack the degenerating peripheral nerves. It's on microglia, and it's also in an endothelial fibroblast-like cell at the blood-brain barrier. So I'll show you some detail about those findings. Next slide. So at the Mayo Clinic, 132 autopsies were studied, both from familial and sporadic cases. The familial cases included SOD mutations, C9orf72, and a couple of other genetic forms of ALS. But the predominant group studied were what we call sporadic, meaning there's no familial association. And what was initially very prominent was that alpha 5 integrin was only in the motor areas of the spinal cord, in the anterior horn where the motor neurons are, and there was a real paucity of alpha 5 in the dorsal column where the sensory tracts are. And I'll show you further pictures, but I asked Doctors Romer and Dixon if they could find an example of a phagocytic cell that was attacking a motor neuron, and they came up with a picture. And I think it would be appropriate, looking at the slide on the right, the photomicrograph is showing a cell eating a motor neuron, or about to phagocytize a motor neuron about 5 times its diameter. So it's fair to call that cell, even if it's a microglia or a macrophage, it's hard to discriminate. It's fair to call it a big eater. It's eating something bigger than it. And then after the motor neuron is phagocytized, there's a debris field. And the area is called a neuronophagic area, and it's full of alpha-5 staining debris. So alpha-5 is not on the motor neuron, but it is on the cells that phagocytize the motor neuron. And there must be signals that are informing the phagocytic cell that the motor neuron is in danger and the phagocytic cell is removing it because that's what its program is telling it to do. So the simple idea here is if we could stop the phagocytosis of a motor neuron sending out a danger signal, then the motor neuron would survive and the person with motor neuron disease could live a lot longer and potentially retain their muscle power. So this is in cancer, we want to inhibit the don't eat me and allow the immune system to attack the cancer. But for our precious motor neurons, at least philosophically, what the bargain would be is let's stop the phagocytic cells from destroying the motor neuron and increase longevity and strength. That's the simplistic thinking. Next slide. So here are some of the pictures. The paper was published in PNAS about 11 months ago. So first of all, alpha-4 integrin is in panels D, F, and H. And you can see the boundary between the posterior column and the ventral horn. So alpha-4 is quite prominent in the sensory areas. It's even there on normal myelin. That came as a big surprise to me. I didn't know that. But alpha-5 integrin is the inverse of that. It's only in the motor areas, and it's on both foamy macrophages and foamy microglial cells. They're called foamy because they look like they have ingested a lot of fat, and that allows the neuropathologist to give it that description. So very big difference between alpha-4 and alpha-5 integrin in human ALS in its various forms. Let's go to the next slide. So the expression of alpha-5 integrin is specific to ALS and specifically restricted to the motor areas. So here again in J and K, K is just a bigger picture of that cell that is phagocytizing a motor neuron, and L and M are the debris fields. When we go, I asked Dr. Romer, can you see evidence of alpha-5 integrin in peripheral nerve? So she did a very clever experiment. Most of our nerves are mixed, but as they come off the spinal cord, the nerve roots, the ventral nerve roots are pure motor, and the dorsal nerve roots are pure sensory, and then they integrate into the nerve so that most of our nerves have both sensory and motor components. And she only saw alpha-5 in the peripheral nerve coming off the ventral root. So that's the essence of the pictures in V, W, X, and Y. And then there's a strong positive staining on IBA positive microglia, and also this could be quantitated. That's shown in U. In the motor cortex as well, there's prominent alpha-5 staining, and that's shown in panels N and O. So the term spatial zonation is very popular, but this is very stark in a very large sample of post-mortem material studied by our colleagues at the Mayo Clinic. Next slide. So in the human post-mortem studies, as I mentioned, they looked at, looking at the left bar graph, non-motor neuron disease, doesn't have much alpha-5, but in the specimens with the SOD mutation, the C9orf mutation, sporadic forms, and then two other genetic forms, TBK and TARDBP, you see much more prominent alpha-5 staining. And then in the SOD G39, G93A mouse, as disease progresses, there's more and more alpha-5 present in the spinal cord. The next slide. So there are two antibodies that we tested. This was begging to be tried after we saw the spatial zonation. MFR5 came from Tim Springer's lab in the 1990s, and 339.1 is the mouse surrogate of volociximab. So we studied the effect of these antibodies in the SOD model. We first did a study at Stanford. That's what the movies that I'll show you were taken at Stanford. And then after we licensed it to Pasithea, they have had the work independently confirmed at a CRO. Then Brad Turner worked on the TDP-43 studies, as well as we had some studies done at QPS, the CRO who did the 3 studies outside of Stanford. And in these studies, we found that twice weekly injections, it was given intraperitoneally rather than intravenously. And we began treatment at 8 weeks of age. The animals become clinically evident motor neuron disease at around day 100, and then they live to be about 170 days, and I'll show you the data on both survival, electrophysiology, looking at the compound motor action potential, beam walk, and grip strength. Next slide. So these are some of the data, the beam walk at 12 weeks. This is measuring slips and speed. So the mice getting the isotype control at 12 weeks had many more slips than the mice getting the anti-alpha-5. Survival was what I would call modest. The p-value looks good. The hazard ratio looks good. And it's 10 days. What 10 days means, if one wanted to extrapolate to humans, is impossible to say. We also noticed that even though in the 2018 paper, we showed that alpha-5 is not on T cells and B cells in the mouse and very little in the human, we were showing that there was a reduction in T cell infiltration into the areas where the motor neuron disease was. The next slide will show what the reduction looked like. So the main effect was on CD4 cells. It affected FOXP3 cells as well, but there wasn't any significant effect on CD8 cells. So it's something to remember. And there was a paper that was published in PNAS in 2019 showing that in the SOD animal, if you take CD8 cells, the CD8 cells will kill motor neurons in culture. So there is a role for cytotoxic CD8 cells in this disease. Next slide. So the three studies done at the CRO on the beam walk, again, there was less slipping and more efficient walking along those beams at week 12 and week 18. So it was highly reproducible. The next slide. And electrophysiology was done, and this was in the, there were two TDP models. You can see them there. So the short model showed that there was, with the therapeutic monoclonals, the MFR5 and the 339.1, there was an improvement in latency and an increase in nerve conduction velocity electrophysiologically. And then at least in the males, in the longer model with the TDP mutation, there was an improvement in grip strength. Next slide. These are just some movies. This is an isotype control. You can see it's dragging its hind legs. The next slide is an animal treated with the antibody. It's a well-groomed mouse moving along well. Then 14 days later, here's one of the few surviving isotype control animals, pretty sick. And an example of the next movie of a group of healthier mice that got the anti-alpha 5. So for a while, we were very thrilled. There were periods where very few anti-alpha 5 animals were dying, but eventually even they hit the wall. So I have some ideas, obviously, about how to improve on what we've been able to see so far in terms of survival. Next slide. And then alpha-5 expression labels microglia and macrophages and increases with disease duration. You've heard me mention that a few times. We also, in the mouse, were able to do CyTOF, and in the late stages of disease, pro-inflammatory cytokines, including TNF-alpha, are present in the alpha-5 microglia, and in the sciatic nerve, where it's not microglia but macrophages, there's a prominent increase in IL-6 and TGF-beta that you can see in the CD49E. Next slide. So again, the survival results, there's some success. It doesn't really change onset of disease. They all start getting sick around day 100, but the progress of the disease really changes until at the end there is all the animals are dying. So one of the problems with giving a biologic, and when I'll show you the PK PD data in the next slide, I think. Next slide, please. Not yet, but we have different antibodies that MFR5 in a fibronectin blocking assay. Alpha-5 is called the fibronectin receptor, or fibronectin is the alpha-5 receptor, depending on how you want to look at it. So 3391 is more of a complete blocker. MFR5 is more of a partial blocker. And we see this in cell adhesion assays. Then the pharmacokinetics is on the right. So it's well known that when you give a monoclonal antibody, that only a small percent gets across the blood-brain barrier. It's too big. Single 5 mg per kg IP injections on the left axis is serum concentrations in micrograms per ml. on the y-axis on the right is CSF concentrations in nanograms per ml. So you can see that it's less than 1%. Multiple injections, we could get doses into the microgram range, but it's still much less. So one of the ideas that I'm working on, I'm spending a lot of time trying to develop a chimeric antigen receptor for alpha-5 beta-1, because a CAR T is going to be able to get across the blood-brain barrier. And so I'm involved in two big grant efforts. One is to make a conventional type of CAR T using manufacturing of a person's cells outside the body, but another is a more modern technique of using mRNA technology just to inject a lipid nanoparticle and see if we can transform CD8 cells to become chimeric anti-alpha-5. So that's one of the aspirations, because before pulling the trigger in humans, it might be reasonable to see how much we can improve the results. Let's go to the next slide. So again, alpha-5 blocks myeloid cell infiltration in the SOD mouse. We saw that again, both in the IBA positive microglia at the end stage, and then also with muscle, looking at macrophages infiltrating the muscle. We were able to reduce those alpha-5 phagocytic cells, both in muscle and then in the CNS in microglia. Next slide. So looking at biomarkers, we did not hit a statistically significant reduction in neurofilament light chain. There's a small trend, but you can see both with MFR5 and 339, it's rather modest at 20 weeks. So right now, in the drugs that we have available, Realiotech does not lower NFL. It's approved. Radicava does not lower NFL. It's approved in some jurisdictions, not in Europe. And then we have the story with tofersen, the SOD antisense, which is approved. It does lower NFL, but it didn't meet its primary endpoint. But the FDA, and I think it was a wise decision, approved tofersen. So NFL is still a commanding choice of a biomarker that one wants to follow with any experimental therapy. But there are some biomarkers that may be more specific for this type of therapy. So I mentioned that fibronectin is a binding partner of alpha-5, and so is a molecule called osteopontin, which we've been studying in our lab for about 25 years in MS, but others have studied it in Alzheimer's disease and in ALS. So I'm going to show you some intriguing data. We're going to do further biomarker studies in the next set of experiments looking at what we're doing to osteopontin. Osteopontin doesn't get a lot of the attention that a cytokine might get, but it's a fascinating molecule which is binds. It's an integrin, small integrin binding molecule. So let me show you the data in the next slide. We have some studies on anti-alpha-5, reversing inflammation and sealing the blood-brain barrier. Next slide shows a HUVEC assay, human umbilical cord vein. So we stimulate with TNF-alpha, the electrical resistance, or in this case, it was dye leakage increases. And if we come in with some of our humanized anti-alpha-5, we're able to seal the blood-brain barrier, so to speak. That's with the AM17 and the A2M5. Next slide. So at the blood-brain barrier, there are these perivascular cells that show alpha-5 CD49E immunoreactivity. We go to the next slide. And there was a paper on the right from Sweden. Altered perivascular fibroblast activity precedes ALS disease onset. It was a letter published a few years ago. Over on the right is just some of the work that we had done over the years on osteopontin. And in this paper that I'm referring to, first author is Anna Manberg. There's a wonderful story emerging that these fibroblasts at the blood-brain barrier are rich in osteopontin. And then at the bottom of the slide is a paper from the Biochemistry Journal showing that alpha-5 beta-1 integrin binds to osteopontin. So let's go to the next slide. So these are some studies of SPP1 is the official name of osteopontin, is sialophosphoprotein 1. And you can see that over at the top on the right is as disease progresses, there's much higher levels of SPP1 or osteopontin in the plasma. And it's really worth taking a look at that paper. So that shows that it's certainly a biomarker we want to follow in this alpha-5 therapeutic. The next slide. It's another paper about SPP1 that was published in Neurology, Neuroinflammation in 2022, showing that SPP1 is elevated in ALS. It's also elevated in Alzheimer's disease, and there's some interesting work there, and then in frontotemporal dementia. So that arrow refers to SPP levels were significantly higher in patients, in sera from patients with ALS compared to healthy controls. And then the levels continue to increase as the ALS FRS score goes down. So it's a very appealing biomarker to look at because of the biology of integrins, and it may help us as we design clinical trials to follow not only neurofilament light chain, but SPP1. Next slide. And that was, again, the negative correlation between the levels and the ALS FRS scores. Next slide. So just to summarize, there's striking zonation of this integrin in motor areas in the central nervous system and peripheral nervous system and postmortem tissue. Anti-alpha-5 treatment in the animal models that we've tested consistently showed survival benefits and motor strength and beam walk, grip strength, and CMAP. So that's the case that the experimental evidence is showing us so far, that this may be a direction worth pursuing and at some point taking into a clinical trial. So I welcome any questions, and I appreciate the opportunity to talk to you. Go to the last slide. We have a couple more clinical plans for the anti-alpha-5 therapeutic. Next slide. So again, we've developed these antibodies, M17 and M5, that have they're fully humanized and we're trying to raise funds to develop a humanized antibody that is based on these specifications. We're going to do that with the CRO. The next slide. All of these human antibodies have an effect on inhibiting binding to fibronectin and reducing phagocytosis. Next slide. We've done cryo-EM, and we know where on alpha-5 our human antibodies are binding, and it's in the propeller domain. So again, you can do a lot with CROs, including cryo-EM. Next slide just shows some of the binding sites on the propeller region. So those are very exciting pictures at 3 angstroms of resolution. So the first type of trial will be done with the monoclonal, but eventually I like to try a CAR T in this disease, given the really remarkable results that we're seeing in some other neurologic diseases, because the CAR T, unlike a monoclonal, will get across the blood-brain barrier. Next slide. Again, the summary, I've given you the summary probably more times than you need to hear it. And now I'll turn it open to questions. Thank you, everyone.
Thanks so much, Larry. That was a fantastic presentation, and really insightful in terms of telling us about the history and how you've come to this point of almost getting it to a trial. I would like to open the floor to questions now. So just a reminder, if you would like to ask a question, we do have a chat available, so you could type your question into the chat and we can read it out for you, or else use the reaction button to raise your hand and we can unmute you and allow you to ask your question.
The other thing I want to say, I'll answer the questions as best I can, but if you're whether you're satisfied or dissatisfied, if you want to have a further conversation when I get done writing my grants, as Sarah can tell you, I answer emails pretty promptly, except when I don't see things on grant review websites. But generally, I'm accused of spending too much time looking at my emails because I like to do it.
Well, just while we're waiting for, we do have a question. Hang on a second. We have a question here from Joe Nicolazzo. I think you should be on, Joe.
Hi, thank you. Thanks for that wonderful presentation. I was just interested to know if you've considered perhaps bispecific antibodies to assist trafficking across the blood-brain barrier, for example, one sort of to the transferrin receptor, for example, to get it across the BBB or blood spinal cord barrier, and then to the alpha-5 beta-1 to then target the actual target.
Early on in this pursuit, we talked to the team at Denali, because they were in the neighborhood and they have a big stash of cash, but they have their own program. So I think your idea is very good, and that would be the leading candidate for a bispecific, one arm anti-alpha-5, the other arm transferrin, and it would be in some ways perhaps easier than the CAR T play, but I think they're both worthy of attention. One of the problems in the field is there's no shortage of good ideas, including the one you just mentioned. You have to get somebody to write the checks, but I think it's a great idea, Joe.
Okay, thank you.
Hey, thanks, Joe. Perhaps I'll ask a question We have a question here in the chat from Rebecca Sheehan. She just wants to ask, in your mouse studies with the anti-alpha 5 antibody, where do you think the slowing in disease progress is coming from? Have you investigated the pathology of the motor neuron body or specifically the axon myelination or the neuromuscular junction, for example?
We haven't done those things, and they're all places where there could be an effect because this is a biologic. Some of it is getting in, but the predominant effect may be out in the periphery because most of it's out there in the periphery. And my attitude is whatever helps would be good. But I'm very concerned about biologics in general for CNS diseases. Alpha-4 for MS had a totally different strategy. We wanted to keep the immune system out of the central nervous system, and we didn't need to get it into the brain. But here, we really would like to see it get in there. So, I mean, there are ways we could try to put it in the CNS, but because of bulk flow, the CSF turns over so rapidly, it just seems like you're literally paddling upstream, and it's not going to work very well.
Thanks for that. I just had a question about the CAR T that you had mentioned briefly at the end. Just tell us a little bit more about whether CAR Ts have been used in sort of other indications and sort of how they're proposed to help with that blood-brain barrier penetrance.
Right. Well, the T cell is very good at getting across the blood-brain barrier, as we learned from the studies leading to Tysabri. So it keeps T cells largely out of the CNS, but otherwise the T cells involved in immune surveillance, and it goes across the barrier. So do B cells. They also have alpha-4. So the idea would be the CAR T is rich in alpha-4, will go across the blood-brain barrier as a normal CAR T, and then as a normal T cell. And then instead of having a T cell receptor, we put in the antibody that will specifically bind to the alpha-5 beta-1 cells at the site of disease. That's the aspiration. But we're making one so that we can test it in the SOD model, and we'll compare it directly with the monoclonal antibodies. And there's some ways of making the CAR T stronger by using a molecule like FOXO1 to bolster the CAR T's ability to fight without getting exhausted. There's this phenomenon of T cell exhaustion. So we could engineer a more aggressive CAR T for a disease like ALS.
Yeah, great. Yeah, we have a question here from my colleague Davo here. Go ahead, Davo.
Yeah, thanks, Sarah. Davo from FightMND. Hi, Larry. Wonderful presentation again. Terrific to see how the progress is coming along with the alpha-5 beta-1 integrins and fighting MND. And really glad to be in position to have supported this. I've really, again, from my anatomy background, really fascinated by your images, pathology images that you showed of the macrophage, which was a beta-1 positive, eating up the motor neuron, or next to and in the process of eating up a motor neuron. Just looking at a biological question in terms of when the monoclonal is added, is the activity there reduced of the alpha-5 beta-1, or is it preventing it from actually having the active of eating up the motor neuron?
Well, both could be happening. Some of the in vitro studies show that actually it's sterically interfering with binding to the motor neuron, but it's also reducing phagocytic activity. We had some phagocytosis assay, which at least shows that. It's also decreasing inflammatory cytokines associated with those phagocytic cells. So those are the in vitro analyses. But I think all of them could be coming into play, and they all could be useful.
And just to follow up on your data on survival as well. You mentioned you were talking through to the two antibodies not having an effect on CD8 positive cells. Is there thought of doing some kind of combination, or is there another integrin that could be involved in reducing the activity of CD8 and then having a combined therapy?
Yes, I mean, I think that this could be a very important addition. Generally, because the costs of getting into the clinic are so high initially, I want to try something and keeping it simple. But some of the ideas, even what we heard from Joe earlier on of putting a transferrin receptor on it, it may be a good strategy. One doesn't necessarily need to have a CAR T. So far, the trans strategy hasn't worked that well in the clinic, but we'll see. Denali and others may come up with something that is just right. So we have to just keep an open mind. The trouble with all of this is we're all real serious about wanting to get this into the clinic and do it safely and maybe make an impact. But there, in my opinion, pharma has been enamored and looking at the specific genetic forms and shutting down the mutant genes, antisense being one of the most prominent. But this isn't going to help people who don't have one of those mutant genes. So I understand their strategy, but we'll see what we can do to get this therapy for the 90% or so who don't have one of the genetic forms. It would also be helpful in the genetic forms because even something like tofersen didn't hit its primary endpoint, although there was a signal, a trend anyway.
Right. We have another question from Joe Nicolazzo again. I think your camera should still be enabled, Joe.
I liked your other idea so much. You want to try for two now? I think you're
sorry,
muted.
just
You're muted. I think they're going to do that during the presidential debate on Thursday between Biden and Trump.
Perhaps, Joe, if you wanted to quickly type it in the chat. I'll just quickly read a question from the chat while Joe's doing that. There's a question that I'm really interested in the data that you showed where in your anti-alpha 5 antibody treated mice, you reported a decrease in CD4 and FOXP3 positive TREGs, in contrast to evidence, particularly from Stan Apple's work, that TREGs can be beneficial in SOD1 mice. Do you have any ideas around this observation?
Yeah, I think Tregs could be very important. I'm rooting for the Koya trials, just like all of us are. So, it was a disturbing fact that we were also reducing FOXP3. So I'd like to see FOXP3 going in the other direction. But I think that we'll get to an answer. Stan is advancing now to a placebo-controlled trial, as I understand. Do you want to give it another go, Joe?
I can try.
Oh, there we go.
Oh, good. We hear you.
Yep. Now I forgot the question. Oh, it was around. So you noted the integrin around the perivascular macrophages in the brain microvessels. Have you seen the same around the spinal cord vessels and even the meningeal macrophages? Has that been looked at?
It's been looked at, not in as much detail. I'm going to let Mayo Clinic know that you asked that very good question, and hopefully they'll have a look. They've just been marvelous. They don't stop working. Very impressive group.
Okay, thank you.
Right.
Thank you.
Well, I think we might wrap it up there for the session. Thank you, Larry, so much for taking time out of your Monday evening there in California to present to us today. It's fantastic for our researchers here in Australia, particularly to hear from an international leader in the field. So I really look forward to seeing what are some of the outcomes of your research are.
Thank you very much. And I also really want to thank the Fight Motor Neuron Disease people for supporting the work. This will always be remembered if we get to the some success. But thank you very much.
No, absolutely. We're very proud to support your work there, Larry. So if there's anything you would like to, if anyone has any further questions, I'd like to follow up with Larry. You can get in touch directly with him, or if you'd like us to help you connect, please get in touch with me, and very happy to help connect you with Larry for further discussion.
It's pretty easy. My last name at stanford.edu. Thank you, Sarah.
No worries. We have recorded this seminar today, so if you know anyone who was not able to attend live but would like to watch this seminar, please let them know that it is available on our website in a few days' time. So again, thank you everyone for joining today. We hope you've enjoyed it. Please join us again next month when we'll have a new speaker. Keep an eye out in your emails for news on this seminar, which will take place on Tuesday, July 23rd. Thank you, everyone. We'll see you next time.